So the purpose of this video will be to go over the soil assay lab and talk about the procedure for the lab and some other pre-lab information. What you should do is you should first read through the printed procedure that I gave you in class and have that out along with you while you go through this video. So in this lab we're going to conduct 4 different assays on soil, and our soil sample will be taken at Richview just outside of our classroom. So the 4 assays that we're going to be doing will be one for the type of soil, the second one will be for the water content, third one is for soil compaction, and the fourth one is for soil pH. The second part of the lab will be for you to design and conduct your own experiment which uses one of these assays, and that will be accompanied by a formal write-up. But we'll talk about that in more detail a little bit later on. The purpose of this video will be to focus on part 1 and these 4 different assays. So when you arrive to class on the day of the lab, please arrive early and ready to work. So make sure you've had your washroom and drink break so that we have the entire class to work. There'll be a quick opportunity for you to ask any final questions and review the procedure for the day before we go outside and actually conduct the 4 assays. After everybody's done outside, we'll all return to class and finish with the final cleanup. So the equipment cart for the day will look like this. Each one of these 4 bins here has the equipment that you need for each of the 4 assays. There's also some other stuff over here which we'll use for each experiment. So I'll discuss this equipment as we go through each of the 4 assays. But at the end of the day, the cart should look exactly like this, and so it should be cleaned up and ready to go for the next class once you've finished with all of the equipment. So for actually collecting our soil sample, we have available to us a number of different shovels and things, and so we can pick these up from the locker that's just outside of our classroom. So we'll use these tools to do the majority of the digging, and then we also have these scupulas which are good for transferring the soil samples into smaller containers like the graduated cylinder. This lab will require you to return the following day in order to collect some data. So I'll be available in our classroom in the morning from 8:15 to 8:45, as well as during the first 20 minutes of lunch, and you have to come with your group during one of those times to collect your data. The reason we're doing this is because some of our tests require that our samples sit for 24 hours before we collect the data. So the first assay we're doing is for the type of soil. So if you read through your procedure, you'll see that the soil is made up of several different components, and so we want to figure out what is the composition of our soil sample. So we're going to take about 30 centimeters cubed of our soil sample and place it into a 100 centimeter cubed graduated cylinder. So something to note here is that 1 centimeter cubed is the same thing as 1 milliliter. So that means if we take a 100 milliliter graduated cylinder and we fill it up to the 30 milliliter mark, we will have collected 30 centimeters cubed of soil. We'll then finish filling the graduated cylinder with water, cover one end with parafilm, and mix the water and the soil together before we leave it to sit overnight. So in class, your goal will be to get up to step number 4. We'll then leave our sample overnight and return to class to do numbers 5, 6, and 7. So when you're done collecting your sample and you have it mixed in the graduated cylinder, you'll just put it back onto the cart so that we can roll the cart back into the class at the end of the day and everybody's samples will be there and ready to go. So just simply go up to step number 4, place your graduated cylinder back onto the cart, and then you can move on to the next assay. But don't forget to return the next day to record your data. So parafilm is what we're going to be using to cover our graduated cylinder. And so parafilm is essentially just a thin, stretchy film that we can use to seal up containers. And so here's a demonstration of what that looks like. So once you have your graduated cylinder covered with the parafilm, you can mix the soil and the water together and then let your sample sit overnight. When you return the following day, what you'll need to do is take your graduated cylinder and measure how much of each layer you have in your sample. So what percentage of your sample is full of silt, what percentage of your sample is full of clay, and what percentage of your soil sample is full of sand. And then once you have calculated that, you can use this table, which can be found in your procedure, to determine the type of soil that you have. So if I have, for example, a soil sample that has 60% clay, 30% silt, and 10% sand, that means that it's a clay sample. So I just look for the place where all of these lines intersect, and that tells me the type of soil that we have. We can just discuss this in more detail when you come back to collect your data. The second assay will determine how much water we have in our sample. So what we need to do is first find the mass of an evaporating dish. So if we go back to our cart, the evaporating dishes are these containers right here. So they're just a shallow dish that are heat resistant, so we can put our sample in there and bake it into the oven. So we first need to take the mass using one of these scales of our empty dish before we fill it with our soil sample. After we have filled about 3/4 of the dish with our soil, we'll remeasure the mass, and that will let us determine what is the mass of our soil before we remove all of the water. That will be the only thing that we'll do in class. After step 3, what we'll do again is place our sample onto the cart and we'll leave it there. When we come back into the classroom at the end of the day, we'll put all of our evaporating dishes into an oven and we'll set the oven to 100 degrees Celsius and leave it overnight. That will ensure that all the water evaporates off so that when we come back to collect our data the next day, we can remeasure the mass of our evaporating dish and figure out how much water content had evaporated off, and that will tell us the percentage of water in the original soil sample. So once again, after we filled our evaporating dish and after we've measured the mass, we can just simply place it on the cart and we'll leave it there until we come back to class. When you're leaving your samples on the cart, it's a good idea to place a piece of tape on them with your group members' names or your initials, just so that you know which sample belongs to which person. So this video just shows you a quick demonstration of what it looks like when you're using the scale. So we first take the mass of an empty dish, and then we fill the dish with our sample. In our case, we'll be using soil as our sample, and then we can take the mass again, and by subtracting the two, we can figure out how much our sample will weigh. The third assay that we're going to do is soil compaction. And so the equipment that we'll use for this lab will be a spool and a knitting needle. This diagram here is a bit misleading. What we're going to do is we're going to place the spool flat onto the ground, and then we're going to put the knitting needle down through the center. However, we're not going to push it into the ground right away. We'll just simply place the needle into the spool until it hits the ground. We won't force it into the ground yet. Once the needle has hit the ground, we're going to mark that spot with a permanent marker. And then we'll go on to the next step, which is to push the needle down as far as it will go, and then mark that spot again. Once we remove the needle, we'll look at the place, the 2 marks on the needle, and that will tell us how much of the soil has been compacted. Something to note when we're working with the permanent marker is, of course, there are many other groups that need to use the same knitting needles. So what we need to do is take a small drop of ethanol and place it on a permanent marker so that we can clean it off so that the next group can go ahead and use it. So the ethanol will make sure that the permanent marker comes off of the knitting needle so that it's clean and ready to be used by the next group. So if we go back to our equipment card again, you'll see that there's a bin that contains the knitting needles, spools, and a bit of ethanol. The final test that we're doing is the soil pH test. So pH is something that you actually will learn about more in grade 10 when you talk about chemistry, but for now we'll just be doing a general overview of our pH level in our soil sample. So the printed procedure that you have gives you a bit of a description about pH and what types of plants prefer which types of pH levels. So I would definitely recommend that you give that a read through. So for this assay, what we need to do is fill one of these screw cap containers with 10 milliliters of our soil sample, and then add 15 milliliters of distilled water to the tube. Cover and shake for 2 minutes, and then let the soil settle for another 10 minutes. So when you're doing this assay, we can do the same thing as the other ones. We can add our sample, shake it, let it sit on the cart. Once you're done that, you can take a pH strip and you can dip that into your sample, and that will tell you the approximate pH value. So once you've dipped your pH strip into the sample, so that's what this is here, you'll see that on the cart we also have these containers that have a color chart on them. Notice that the color chart and the container are inside a plastic bag. Please leave them in the plastic bag just so that they don't get damaged. So you can compare your pH strip to the color chart on the container, and that will tell you the approximate pH of your sample. Again, please make sure you read through the printed procedure so that you have an idea of what the different pH levels represent. So we'll just go back to the equipment cart one more time. Your pH strips will come from this container right here, and these plastic bags here have the color charts. So don't take any pH paper from these, only take your pH paper from this container here. This is also where you'll get your parafilm for the graduated cylinder, and then this container here contains a bunch of Sharpie permanent markers that you can use to mark up your knitting needles. When we return to class, we'll return our digging tools to the locker, place our samples in the oven, and then make sure that the cart has been reorganized and put back to the way that we found it so that the next class is ready to go. So that will be it for our pre-lab talk. Make sure that again you read the printed procedure through so that when you come to class you're ready to start working on your lab.